Hello everyone and welcome to today's career development session, today with a special talk prepared within the SNEV mentorship program. My name is Helena Branco. I'm a PhD student at both University of Porto in Portugal and University of Torino in Italy. And together with my colleague Carlos Jesus from the University of Coimbra, I will be moderating this session. So it is my pleasure to introduce today's speaker, Dr. Diana Jovanovic-Talisman, an associate professor in the Department of Cancer Biology and Molecular Medicine at the Beckman Research Institute of City of Hope in California, United States. Dr. Diana received her bachelor's degree in physical chemistry from the University of Belgrade, Serbia, and her PhD in chemistry from Columbia University in New York. She subsequently completed a postdoctoral fellowships at both the Rockefeller University and the National Institutes of Health before establishing her lab at City of Hope, where her group is developing advanced imaging platforms to quantify single extracellular vesicles. So in today's talk, however, Dr. Diana will focus on how to transform messy or complex data sets into clear, compelling, and scientifically rigorous figures, sharing with us some practical strategies for improving visual clarity, simplifying data presentation, and strengthening scientific storytelling, which is very important. So before we begin, as always, we'd like to encourage you to please post your questions in the chat box during the presentation, or otherwise to unmute yourselves at the end of the session in the Q&A to speak directly to the speaker. So now I warmly welcome Dr. Tiana to start her presentation. Thank you so much.
Thank you so much for this really kind introduction. And thank you so much for having me, because this is something I feel extremely passionate about, right? We are scientists. We love being in the lab. We love being next to the machines. We love seeing our data. And sometimes the data is so clear to us. And it's almost a trouble to understand that not everyone sees it immediately. So I try to translate that excitement that I see when I see the raw data into my figures. So I will tell you a little bit about how do I do this. And I think the end result is really amazing. You know, you start by struggling because it takes time, right? You learn programs, you learn how to put data together. So it is time consuming. But it's something that really translates, you know, even beyond the science, how you approach your interviews, right? How you talk to others, how do you put these complex pictures into beautiful figures. So I'm going to share my screen right now. And okay. And share. And we'll start it. Okay, so do you guys see it? And do you see it in a good, not presenter mode, but you see the full screen, right?
It's in presenter mode now.
Okay, excellent. And it's great because, you know, no matter how long we are in this kind of half virtual world, we'll still struggle to make it easy and always need to check. Okay, so as you heard today, I will tell you about making figures pretty.
I'm sorry. It's in presenter mode, I believe.
Okay, so I swapped it a moment ago, but let's do it again. Is it good now?
Perfect.
Okay.
Thank you.
It's funny because the moment ago we tested it and this worked, and now I have to do it again. So, you know, good. I'm glad we checked. Okay. So, as I said, I will talk to you about presenting your data and making figures that are not only pretty, but represent your data well, right? So I will tell you about how to tell your story effectively, and I will start with my first scientific story. So this little bit dates me, right? This is my first paper. And I did it while I was at the Mayo Clinic in the last year of my undergraduate training from the University of Belgrade. And I entered this amazing world of science. I entered data, exciting data. And then there was the point where I needed to make that pretty data into words and into figures. So we made this beautiful paper that was part of my thesis. and I translated into Serbian. So I had my thesis in Serbian. So my mom was proofing this. And I was really excited. I was like, Mom, did you understand what I did? And all of things that she understood is that my protein was blue. So unsurprisingly, my mom had this whole thing going on for my thesis presentation. So after my undergraduate thesis, she had a blue cloth and blue plates and blue cups, right? So it was lovely. But I learned at that moment that perhaps my figures were not as clear as they were in my head, and perhaps I could work a little bit on my presentation. Now, I also wanted to say that from the moment that this paper was presented, things have changed quite a bit. Graphical programs were scarce. Presenting data in colors was really still a challenge because you had to pay extra for color figures. You really had to pick carefully how to do things well. And we grew significantly. So I want to incorporate that kind of growth into my talk. Okay. So I wanted to start telling you about programs that I use. Now, this is not an extensive set of programs. This is something that I am comfortable with. Doesn't mean that you should be, but I'm just going to tell you what I found works really well. So obviously, when you make figures, you have to think about a process. The first thing is that you compile your data in a program. And then you look at, statistical data analysis, and then you make a figure. So for this first point where we put data into kind of understandable set of numbers that we can interpret, I use Excel or Prism or MATLAB. I find that Prism works generally really well for our data because it is allowing us to do statistics immediately, make pretty pictures that can be translated, with graphs can be translated. However, I want to warn you about one thing that we did kind of encounter. Prism has a limited number of data sets that you can put in. So sometimes you can get into trouble. So we worked at extracellular vesicles, right? Sometimes there's so many of them, and Prism can make trouble with that. So I'm just kind of giving you tips and tricks that we found along the way in our lab. And when I make schemes, I typically either use Adobe Illustrator or BioRender. Now, BioRender is super easy. It is free for many people to use, at least in this kind of limited version. So it does work really well. However, the way you can design your data is somewhat limited. Adobe Illustrator, on the other hand, is first a pretty pricey program, but it allows you to make your data really nicely edited and really highlight what's important to you. It's a steep learning curve, but in my lab, I always emphasize that that's something that's investment in the future because it will kind of carry, right? That's one of those programs that you can use for many different things. So if you have an option and opportunity, I strongly recommend using Adobe Illustrator. Now, I will talk about something that we all use, but always struggle and think about what's right or what's wrong, using AI platforms, right? So I am very careful when I use AI platforms. I think that you all still need to be use them right, use them appropriately, and use them for the right thing. So you can make amazing, beautiful schemes that are made with AI platforms. Now, you have to be very careful to check if you're submitting a grant. Figures that's generated in AI platform is not really accepted. If you're making a presentation like I am making today, I will show you a graph that I generated using an AI platform. You need to check it, right? You need to make sure that you go through it, that your thoughts are translated there. So it's really not that AI platform makes the scheme, you tell it how to make it, but you need to check it and you need to disclose. And finally, I use Adobe Illustrator or GIMP to finalize graphs, especially for consistency and alignment. So I have to say that when I make my figures, it's super important for me that everything is aligned. Why? Because if everything is in order and everything is aligned, then I will not pay attention to misalignment is important. I will pay attention to data. And my whole talk really will focus on making sure that you pay attention to the data. Okay, so what are things to consider when you make figures? Make important information stand out. That really kind of connects to my previous statement that you do not want to see the misaligned bars or titles or numbers. You really want to think about the actual data in front of you. You want to reduce the clutter and be consistent. Again, that's because you want your data to stand out. If there is a lot of clutter, your eyes will not be able to focus on what really matters. If you're not consistent and changing the font type or font size or font alignment, again, your eyes are going to be going to something that shouldn't be there. The next point is perhaps counterintuitive, but it's really important. And I always start with gray. That means that I don't make my axes black, my font black. I like to make them gray. So my students know this really well in the lab, because if we start with gray and not black, we will not focus on axes. We will focus on data. Black is really strong color. It draws your eyes. You do not want to do that with axes and with your titles and numbers. You want to point to the data. So I tend to make my graphs start with gray, then add splash of colors. And I always play with that. And I show my students often the differences. And it really changes the perspective once you see how it works. This really important scientific point is to use optimal graphs. So for example, we need to know when to use bar graphs. Bar graphs hide individual data points. Is that something we should do? Perhaps not, right? If you have points, you can use graphs, but add your data, add your points, because there is a wealth of data in these points. There is heterogeneity. There is there is how much your data varies, right? You want to show all of that. So bar graphs are not always the best practice, and we'll talk a little bit about that. But sometimes there are multiple ways you can show your data well, and then you kind of need to think about it. What is your take-home message? What do you want to point out? Important point that comes next is that you want to optimize your color combinations and fonts. Why? Because a lot of people, about 10% are colorblind. We want to share our data with everyone. I learned this lesson early when I was designing my figures as a microscopist, right? We'll tend to put green and the red in two different colors because that's how we think about the data. And then I showed my picture to a colleague who couldn't see well the differences between green and red because it's a particular kind of a color blindness. So I learned very quickly that green and red are not optimal color choices. And I want to be inclusive of everyone understanding my data. And finally, we really want to optimize our figure panels because if they're misaligned, if they're not designed well, we want our data to shine. We want to have white space because it's good, but not too much. Okay. So let's start with what stands out, right? Here, it's super easy. You have this beautiful red flower. It stands out in this sea of purple. It's easy for us to know that this is what we should focus on. But what if we have something like this? What's the first thing that you see that's different? Well, for me, this little L that is in pink color that's twisted, it stands out. I see that really easily. It takes me a second to see what's different there. But do you see anything else that's unique here? Now, this is kind of a point I was saying earlier. This would be a lovely talk if I could have an audience in front of me and we can discuss it. So I kind of can give you a hint. But there is one T that is different than all other T's. There is a green T. It's also unique. As much as this L is unique, it's a single one that's different. But it took you much, much longer to find that second unique symbol in this figure. And this is a beautiful, this is actually a paper. This is a figure from the paper that talks about search efficiency. So it takes you longer to identify this green T and much less time to identify this angled L. So you want your audience to see the angled L, right? So that's how you want your data to be seen. You want your data to be clearly and almost immediately recognized. So how do we enhance our data? Well, the first thing is position, right? So when we have our data in a line, or we have a scatter plot, right? And we want to see how two things correlate. Position shows us important trends. When we saw this type of data, we understand that there is relationship between two axes. That's position way of thinking about how your data is presented. How about the size, right? Size is really important. We see something small and we see something large, and it really shows us the magnitude. If we have something bigger, it immediately comes to us that the magnitude is larger. So you want to use this to your advantage. You want to really emphasize your data there. Shape. Shape is something that we are more accustomed using. So if you have two different categories of data, you will see circles on one end and then triangles on the other, right? And you're communicating that these are two different types of data. You can put them on your graphs in a different manner, right? They can be part of different categories, but it can also be part of a data on a bar graph, right? You can see your data as triangles and squares and diamonds, right? So you have additional way to show different categories. Color. We all love color. The splash of color is extremely important. You can see color beautifully, and it shows groups. It groups very easily for our eyes. We see differences immediately. The line width is something that we can also utilize. If you have a thicker line, you will pay more attention because you're trying to communicate with your audience. This is the one that you should be really paying attention to. So this very high thickness is signifying to the audience that you should be paying attention to this very thick data point. And then finally, line type. If we have a dashed line, we will not take it as seriously as the solid line. That's in our nature. This is what we were taught. And you want to use this to your advantage if you are showing the data in this manner. And again, for full disclosure, this is something that I generated with AI. This is the figures that AI generated, but there was a conversation with AI, and I wanted to explain what I wanted to tell with this figure. So hopefully AI plus me helped you here understand what are kind of ways to enhance your data. And I will show you this in a slightly different manner. I will show you how you can highlight your data in a grid. And this is a grid of extracellular vesicles. There is a scale bar at the bottom. And I wanted to point out that this scale bar is really important. So when you are thinking about presenting your data, make sure that important scientific points are present. At this particular point, that scale bars is really, really significant because it gives you the magnitude of our vesicles that are presented. So Nan in my lab was very kind, and she gave me a bunch of vesicles that I could arrange in the grid and show you how you can see the one that I want to highlight. So for example, I can enclose it. If I enclose this vesicle, your eye immediately goes to it. Another way to highlight an important vesicle is to show the thickness, right? You can make certain points thicker than the others, and immediately they will come up to your eye. Location. I just misplaced one of the vesicles from the grid. Your eye was drawn. That's search efficiency. Think about that. It was immediately clear to you that that's the one that you should be looking. Size and shape, right? This is a very different vesicle in the corner at the bottom. You don't really need more than half a second to notice the one that is unique. Text. Sometimes all we need to do is put a text. This is EV number 4, right? It's highlighted. I know it's there. Color, something that we use very often. So although this is a much lighter color than the black, it still comes up very, very fast. Sharpness. Sharpness is again something that we can say, we can kind of blur the rest of the data, and then the data that we want to emphasize in front of us, and then also saturation. So these are all the ways that you can highlight something of importance to you. So this is an example of extracellular vesicles, but you can apply that to any type of grid or anything else that is important to you. And here is actual world data, right? So this is an actual figure in a paper that we just published. And I wanted to highlight vesicles that have a complex morphology. It doesn't take you much because here I had text, complex CVs. I have different colors. I had magenta and green, and I enclosed them. So there are multiple ways I worked to show you how some data that is of importance can be highlighted. So this is one way. Okay. So let's talk about the actual graphing. So we know how to highlight data, and the first time I used Excel, I made pretty messy figure. And I'm going to show you that reducing the clutter is really important because simplicity actually highlights your data. It is initially counterintuitive, but it's really the case. First of all, 3D is cool. Maybe in this particular case, this 3D graphing of my bar graphs, not necessary. It just distracts me from the data itself. Patterning as well. It's kind of odd. Yes, I see the differences, but could I do it differently? I have grid lines. These grid lines are not extremely helpful. They're cluttering my field of view. I have numbers, my tick marks are very close spaced together. Again, it distracts from the data itself. So how can I make this data simpler? This is the simplest way I could do it. I make my three-dimensional data two-dimensional. I had my bar graphs show error bars. I have tick marks, the good spacings. I have my x-axis. I have my y-axis. So it's looking pretty good. So I have now data that shows different captures of EVs, and I can see how many EVs did I detect. So if I capture with CD9, I have something about 0.36, let's say, vesicles per micrometer square. Okay, now, should all this data be black? Perhaps not, right? I like to start with gray, as I said. So instead of making everything black, I made everything gray. And then I varied the grayscale to have different capture shows at a different grayscale. So I like to use saturation difference of at least 25% as a minimum. So you can see very dark, mid-dark, light dark, and very light. So this kind of differences in saturations are pretty prominent. So your eye doesn't need much time to see the differences in the data. But I wanted to show you that we made it better, but it's not perfect. So you have ugly, you have wrong. Why do we have wrong? Because there is no Y scale bar. I don't understand what that data tells me. What are the values, right? I have bad, you know, I have my scale bar, but I have it right next to each of my captures and doesn't look good. And I have something that's okay, but perhaps I can have something that's good, right? So this is much better. So I made my numbers larger, so they're clearly visible. I reduced unnecessary white space. And I really want to emphasize that white space is needed. Don't kill all white space, but don't make it prominent. And of course, your titles are now nicely capturing what you're trying to say, and they're large, and you know, you have reduced the size of your graph and it still tells you the full story. Okay, so what are the things here that we want to pay attention to? We want to be consistent with font and size, and we want to make sure the data is clearly visible. We want to align the text. If any of these numbers were misaligned, your eyes would notice that, and it would kind of shift your attention from the data itself. And you really want to reduce unnecessary white space, but leave some white space. But here is the point that I started to make earlier. We also need to make sure that we represent the data the best we can. So bar graphs, no matter how informative, are not always the best way to present our data. So here, I captured all the data that I acquired. I'm not giving you the average. I'm giving you every region of interest that we imaged, and we am showing you all the data that's represented here. So I think that really makes a difference. So it's not only what looks good, but how we as scientists represent our data most rigorously. And most rigorously is present every single data that we took, right? And of course, at the end, we can add splash of colors and we can add significances. So this is kind of a step-by-step building up our graphs. At the end, we can see the differences in different captures. We can see that I left my control, my IgG control gray, because I don't want to emphasize my control. I really want to emphasize the differences between capture of EV markers, CD9, CD63, and CD81. And I want to show you that CD9 is more abundant than CD63 and CD81. And now when the significances are there, you really see that story that I was trying to tell you. CD63 and CD81, not so abundant, right? So that's basically the way that I think of building up to a good way of representing a figure. Now, sometimes there are several good graph choices. So let's say that I want to show you, so this is the same data. It's from our Jeff paper. It's from 2023. I'm just kind of showing you differently in building up how we do it. So we can show all the diameters for these 3 different captures, and we can show that as a box plot. So we actually now have our average, our median. We have quartile ranges on the left, and we have these dots that represent vesicles that are above or below the quartile ranges. We can present this as a violin plot as well. Another way to present this data is with histograms. So you can see how beautifully you see the distribution. And it does remind you of these violin plots, as it should. You can, for example, combine this far left and far right and show these box plots together with the histograms. And that's something we actually did do in our work, but these are all the ways that you can show your data. Neither one of these is wrong. It just depends how much space you have and what you want to emphasize. So sometimes there are multiple good choices to present the data. The question is, what is your take-home message? Okay, and then let's talk a little bit about the color. So I wanted to tell you that you should always pay attention to color associations. Why? Because inevitably, I put my IgG as a control in gray because that was not really the highlight of the data. I tend to put my cancer data in red and my healthy data in blue. Why? Because we normally associate colors with certain things. So red is commonly associated with danger. Blue is often associated with calm. And it has its connotations in biology, in cultural conditioning, and it's also opposing colors of the spectrum. So I'm a geek. I'm a proud geek. So I know Superman is not always fully blue, but there is a blue Superman. So I put it there, and Lex Luthor on the bottom in red. So good and bad, right? So opposing colors. But blue and red are possibly two best ways to represent your data because those are colors that are seen by everyone. They're on the opposing colors of the spectrum, as I mentioned. And sometimes you can use them to your advantage to really associate these colors with the meaning of your data. Okay. And I also want to tell you that you should be selecting your colors wisely. Approximately 1 in 12 men and 1 in 200 women have some form of color blindness. And this is really important. So look at this top left normal vision scheme and then see how others see it. So that's why I was telling you the difference between red and green for many people will be very hard to see. So you should be trying to pick colors that others can see. And that's why blue and the red is a really good color combination because you can see that most people can see these differences. So I tend to always think about color combinations in the sense that it should be visible by everyone. So how can we adjust the color? Well, we can adjust the hue, so type of color family that we use. We can adjust the saturation, that's intensity, how vivid or how gray our color is. And of course, we can adjust the lightness, so white versus black, how bright or how light our data is. So these are typical things that you will see in graphical programs, but they're really, really important to highlight your data. So what color combinations can you use? I really want to emphasize that sometimes giving different saturations of gray or different saturations of, let's say, blue is really, really good. It doesn't necessarily have to be two different, a very different color. Sometimes looking at either gray scale or color lightness can be really good. Another very good combination is any color plus gray, right? Again, your colored one will be emphasized by your eyes. That's when you choose some type of data like that. Another good color combination, so for two different opposing data, both equally important, red and blue, as we discussed, and magenta and teal. Now, we get often in a situation where we need to present more than two colors. So these are some of the good combinations that I picked. So red, teal, and gray, blue, orange, and gray. And when it happens, sometimes you can actually have a lot of colors that you need to put together. And rainbow colors are harder to see. These very carefully picked colors by the experts sometimes do much better. And another thing that I want to emphasize is once you have this beautiful panel, is how do you put it together? So this is from Nature Research Group. They had this beautiful way of showing you that panels can be arranged well to effectively tell the message. So obviously, neatly arranging, that's something that's really clear to us. But also think how we read. At least in most Western cultures, we read from left to right, and then next is top to bottom. That's how our brain functions in a normal. But I do understand that not all cultures are the same. So we have to be sensitive to that as well when we present our data. So we typically flow the panels from left to right, followed from top to bottom. So you will not see kind of zigzagging as much unless you have to. And then emphasizing more important panels with size, it's something that I also wanted to tell you that you should be using. Remember that first representation of the graph is showing that the size of size shows the magnitude. So if your panel F is really big, it can be big because you really think that that panel F shows the most important data that you're trying to showcase here. Okay. And then another thing that you should be thinking about is what is the font that you should be doing. So the real important point is the simplicity, again, makes a huge difference. So something like Arial or Helvetica, the sans serif fonts are much better for the figures because they're missing these thick marks at the end of the letter. That's what that means. And these are making figures clearer. So we are just trying to simplify the data, distract from unnecessary details. So you can see how this word extracellular vesicles looks in these three different fonts. And then also we need to consider the hierarchy of the text. The biggest bolded and circled is what our eyes pay attention to. So our contrast ratio needs to be high because you can very easily see stuff on the right, but you have harder time seeing easily stuff on the left because you have the low contrast. And this high contrast is the first thing that you see. Why? Because the font is bigger and it's bolded. So use this to your advantage as you're making your figures. And I have only 2 more slides. We're almost there. I wanted to tell you at the very end, so you made these beautiful figures. And this is, again, I collaborated with AI to make this figure. So we need to integrate take-home message. We want to tell the story, but we also want to say something about the text. So when I put a figure caption, I like to incorporate take-home message in the title rather than just descriptively showing what the figure is telling me. So for example, I wanted to tell you about how EV heterogeneity can be revealed by coated transparents, right? So I can start with EV biogenesis, highlight some of the vesicles that we study. I can tell you that EV in biofluids are very heterogeneous. They have different sizes that we can use something that I love so much, which is nanoscopy or single molecule localization microscopy to analyze this data. We have this beautiful, pretty picture. And then at the end, what is the biological relevance? Because at the end of it, however we turn it, it is so what? Why does this matter? So, we really want to show our data together with this take-home message. And as the very last slide, I'll tell you what not to do. Do not mislead. This is really, really important. And this is 2 nice examples from the figure. How can you be misleading? So if you show relative size using the area versus using the radius, you're really showing differences that are not always being represented the way they are, if that's exactly what you're trying to do. Or cutting your bar graphs. So if you use the full range, these bar graphs on the top don't look very different. But if you just cut this very end and then stretch this data, oh my God, you start seeing these differences, but are they real? So yes, we make these figures pretty. We went through all of this, right? But I want to hone in this last slide and tell you, don't mislead when you present your data. That's the take-home message. Make your data show the beautiful story you have, but show them the right way. And of course, you know, as I was making this figure, I had amazing lab, Mihailo, Diana, Nan, Ben, and Ying Chao, who helped me make these figures, because this is not just my talk, it's their talks as well. And I tortured them quite a bit to make pretty figures, but it really pays off because they often win poster awards. Nan just got a poster awards a couple of days ago. So congrats, Nan. And it's really kind of a build up into them to make these pretty figures that tell their story. So hopefully I inspired you, and hopefully your moms will not only see the blue protein, but will see other stuff in your data as well. So I am now going to stop sharing, and I will be happy to answer any questions that you may have.
Thank you very much for this talk, full of colors and shapes, of course. It was really nice to see some of the tricks. I learned some of them I already use it for myself, but it's really nice to see other strategies to be combined with what I already use. So now we can open the Q&A session. So if anyone has a question, please, you can post it in the chat box, or either you can mute yourself and talk. In the meantime, maybe I can ask a couple of questions just to start the conversation. So I would like to know your opinion in which type of errors do you consider that they are most commonly used, not only in the EV field, because I believe this is a transversal area that can be implemented in science. So which type of common errors do you think, or do you see most often, and how should we address them?
So with figures, the cluttering is probably the most common error that I see, because less is more.
Mm-hmm.
That's what I think, because again, remember how long it took you to search for that second T that was in green. You want to see your data immediately. You don't want to go through the grids of very narrow space with tick marks and grid lines. They don't help you distract from seeing the data. So for me, the thing that I find that's one, and the second, inconsistency, because I pay so much attention to stuff that is misaligned rather than the data. The cluttering and I say inconsistencies, those are things that, you know, obviously now I review a lot, papers, grants, those are things that I see immediately. It bothers, right? Instead of me thinking about science, I'm thinking about misalignment. That's so easily fixed. So take this extra, 10 minutes and align your data. Your reviewers will be grateful. That's basically the bottom point. Your reviewers will really, really be grateful.
So, and a follow-up of that would be, how is your process in the sense to balance when it's too much information for the figure, and also the aesthetics versus the scientific accuracy?
Okay. So aesthetics never win scientific accuracy. So that's like very clear. So I always will go with scientific accuracy and being robust in my data presentation. So that's not a question even. And I think that that's something that you all feel very strongly about. Otherwise, you wouldn't be here listening to this talk, right? So that's the first thing. You kind of need to balance certain things as you're making figures. Sometimes you need to, as you're doing this process, I think you'll learn more about your data. You can represent your data many ways. As you're making a figure, you're realizing what's most important for you, right? Do you need two panels, or everything goes into one panel? And that's a scientific process, at least for me. So sometimes, we all sit down in the lab and discuss our data, and we go about it in the way that brings up the data in the figure, but also clarifies our science. Because sometimes making figure process does teach you how to interpret your data and take it to the next direction.
Yeah, and it's kind of also a work in progress. So it's trial and error.
Yeah,
Sometimes in our head, it makes sense to put the data in a way, and then when we put it actually in the paper, it doesn't make sense. So then we need to adapt, right? So.
and that's a great point you just brought, Carlos. So I think it's also exciting and important to share your data. So for example, I'm at City of Hope. We have a graduate program, and we have Evolving Story Forum where students can present their ongoing stories. And these types of forums, if you can find them in your institutions, are really good, because that's the way to kind of litmus test your data. Is my figure telling the story to everyone or not? So showing your figures in this kind of initial phases is sometimes really beneficial. It'll make you think about how to make figure, how to perfect your figure, but also how to make your data shine, or what's the take-home message of my data. It may even help your science. So I strongly advise you to share your data as you make it with your colleagues, even if it's not at the talk forum, perhaps your colleagues or next door lab, someone that you trust, someone that your lab works well with, maybe they can give you advice. And obviously share your data with your lab mates, because they will be there for you.
Yeah, that was one of the questions that I had prepared for you, that it was how early should we start to prepare the figures when talking, for instance, in the paper, but
very early.
Yeah, right.
Yeah, because that's an iterative process. For me, that's really important to realize that I make my figures and I have talk now, and my talk in 2 months will have a different figure. I will not recycle. It takes extra time, but my thought process have changed. I want my data to shine. So this extra time that I take to make this figure, I think it's well spent.
And what is your opinion about mock figures, in the sense that you haven't started with the experiments yet, but you already kind of see where you want to reach?
Okay, so I have a problem with mock figures, and the mock figure problem in my brain is that you're biasing yourself with what you should expect. So if you can use the mock figure in a way that doesn't bias your view, then that's okay. But often I think when we make a mock figure, we have expectation. And I think that can be a little bit dangerous.
And in the sense of like assembling the figures, even if they are not fully complete, I feel that sometimes it helps to try to understand what is missing, right, for the paper. Like, I need to complement with this type of results of methodology, or even appropriate controls that are missing.
Absolutely. Right? And speaking of mock figures, I sometimes prefer empty panels that will tell me where I write, this goes here, right? Rather than having a mock figure. Again, I just don't want to bias anyone.
Sounds good. Yeah, I have so many questions that I could add, here in the conversation.
I think there are a few chat.
Yeah, exactly. We can go now for the chat. So Akash Modi is asking, while in the process of gathering my data for publication, I fall into the trap of feeling like a lot of my data is supplemental data and not enough of it is a main figure data. How can I determine the balance of main figure, supplementary figure?
So that's a great question. And that's something that comes with experience a little bit. And sometimes if you have really good reviewers help you. Sometimes you get so attached to your data. I know I'm guilty of that. Sometimes I have the opposite problem. I think everything should go into the main figure. And it's because I love my data so much. Sometimes I know how long it took to get to something that's just kind of a control. But I feel very passionate about the process it took. I would say that presenting your data early to your group first, and perhaps to some forum that you may have in your institution, is a good way to determine what's a main figure, what's a supplemental figure. I'm sure that your advisors and mentors can help you with that. And at the end, reviewers are there to help you. And sometimes it's decided for you. For example, certain journals have limits on your figure numbers. So you will have to make hard choices based on the journal.
Yeah, I think this was quite clear. I'm not sure if anyone else have any question. Otherwise, we have here a question that is not related with the talk, but since we have some time, I hope it's fine. So it's from Bhuvan Bhaskar. Dear Tiana, as an early career researcher after completing a PhD, could you please shed light on how we can approach postdoc positions and ensure we get a response?
Oh, that's a good question. Absolutely, I'm happy to answer that. So it's really important that you search for a position that matches your skills. If you do not match it, it's going to be really hard for a PI to respond because they have so many different emails. And if they glance your CV and there is nothing there, likely you will not get the response. However, if you find something that's really a good match, the first advice I have is email PI directly. So don't only apply to the position that HR posts. It's important that PI sees your application, that you stand out. But when you do that, make sure that you write a very polite, small email. You can add a cover letter to it as an attachment. But why would you be a good fit? And how is your previous research connecting to your newly found position? Why you are a good candidate, which skills or which interests do you bring? But most importantly, read your text of the position that's advertised. If there are 3 different points that are clearly highlighted as important, make sure that you do emphasize these points in your cover letter. So I think that those are all important things that I tend to reply to most of the letters that come to me that are in my email. But if it goes to HR, often I don't see it in 2 months. So remember that, and remember that PIs are also busy. If you really feel that you're a good fit and PI didn't respond, it's okay to follow up. I don't see that as a bad thing. Sometimes I will be just slammed and miss it, and it's important. It's okay to follow up.
So a direct approach to the PI, it's always better. And even, for instance, if it happens to be in the same conference, maybe to reach out and ask directly to the person.
So of course, that's the best option. If you know someone who knows the PI, introduction always makes a huge difference. If you know someone, you can even mention them in that email. If your PI knows the person, make sure that PI sends that email, right? So there are many ways that this can work to your advantage, but networking is important in that sense, yeah.
Thank you very much for that. So we have another question from Sirvan. I get the idea that the clean and nice presentation of the data can potentially have a good impact. But does it mean that the cosmetic and aesthetics can cause you to be excluded from a certain journal? It can be more of importance as if you are using different software and plot templates. Some can have more nicely looking plots that I might not have access to.
I don't think that nice plots is what I was going for. I was really trying to go for a clear plot. Show your data. So don't show bar graphs, show dots that represent individual data points. That can be done in pretty much any program, including the free programs. So I don't think that any journal would exclude that. I think that aesthetic matters, but aesthetic, of course, is not what's going to get your paper in. It's the data itself, right? Your job is to make your data shine. You want to emphasize it, but data has to tell the story. So I think that no journal will reject your paper because it's aesthetically unpleasing. You just want to help your reviewers at the process of evaluating your data. You want to make their jobs easy. and fun, right? When I see a good paper with good figures, I notice stuff, and then I can get more constructive comments to people who send that paper, and it's so much easier on me, and that you can do with any program. So it's choosing colors, choosing plots, choosing not to clutter, choosing to start with gray. All these things can be done with absolutely anything. I don't think that any fancy program, yes, I like Prism, I know it's expensive, but you can do that even in Google Docs, in a Google Sheet. So it doesn't really need to be paid for.
So I hope this was clear. I think we don't have any more questions in the chat box. So maybe I can ask just one last question, and then we can wrap up the session. So you emphasize, and I think that's really important, because sometimes we are not aware, or we are not aware of people that has these limitations with color blindness or this kind of limitations. So is there any kind of tool that you use or that you would recommend that we ECRs can use to check the color combination or the colors that shouldn't be used for this kind of to avoid these kind of problems?
Yes, so there are ways on the internet that you can kind of have the lens of someone who has a particular color blindness. But I think that these simple schemes that show you how someone with a particular, I chose 4 most typical disorders, how they see the color will give you a good hint of what you should be using. So that's what many people base their color combinations. So either kind of grayscale, red and blue, and so on and so on. So I think that just understanding what they see is where you can kind of make your color choices more inclusive of everyone seeing them.
Yeah, for instance, something that I try to do is, besides changing the colors, is I try to give different symbols in a way that is clear, but also to help people, even if they don't want to print the paper with colors. That's already a help, in my opinion. So maybe also with this, it can help.
Absolutely. That's a very important point. I think that this is something that I use often, and that's why my first slide was all these different things that you can do to highlight your data. So color combined with different symbols does make a huge difference, because you're absolutely right. Sometimes we just have a great color printer, so make it work if you can. And that's why different hues, right? Even if you're choosing colors, can help you.
Sounds great. Thank you very much. So yeah, I think we reached the end of the session. It was really nice to have you here. Thank you for your time and for sharing your thoughts on this topic. It was really, really great. And thank you everyone for being here. See you in the next session.